Hello, everyone, and welcome to Global IMMUNOTALKs. I'm Asia Rolls, and I'm going to be the host today. It's a great opportunity that we have today to hear two amazing speakers, and actually, so Linfa Wang and Matean, and they're going to give two talks that are going to follow one another. And both are in Singapore, so we want to thank you for joining us at midnight. And we're really excited to hear about your talks today. And as you know, Global IMMUNOTALKs is an international platform that allows everyone to listen to the talks. They are also recorded and streamed on YouTube, so you can go back and listen to them later on. But today, first, I'm going to introduce our two speakers. So first, Professor Linfa Wang, who is a professor of the program of emerging infectious disease at Duke NUS Medical School and is the inaugural executive director of PREPARE, which is the Ministry of Health in Singapore. He is an international leader, Batman, in the field of emerging zoonotic viruses, virus host interactions, and bat biology and immunology, which I think has been transformative to what we understand now. And also, especially in response to COVID-19 pandemic, he has served and still serving on multiple WHO organizational committees for COVID-19. He's recently been appointed as the newly established World Health Organization Collaborative Open Research Consortium, the CORC, which is on coronavirus. So we're going to hear much of the most interesting science that we have today, as Professor Wang has more than 550 scientific publications in some of the top journals. He's been elected as to the Australian Academy of Technology and Technological Sciences and Engineering, and American Academy of Microbiology, and the Australian Academy of Sciences, the Singapore National Academy of Sciences. It's clearly one of the most recognized scientists we have out there today. And the title of his talk will be Bats and Viruses from Outbreaks Responses to Bat Immunology. Then we are going to hear the next talk by Dr. Matean. He is also a professor in Singapore, but in Nanyang Technology University in Singapore. He's been a student. He did his PhD with Professor Wang, but he's also an MD, and he's an internal medicine physician at Singapore General Hospital, and recently actually moved and dedicates more of himself to science, basic research, and he's now an assistant professor at Nanyang Technology University since 25. His work is actually helped to establish bats as a great model to uncover new strategies to fight human disease. And in 2019, he was awarded Young Investigator Award by the International Association for Inflammation Societies. His work, and especially a work that he published in 23 in Cell, actually led to a patent, and then in a company that is seems like he's doing very successful, and actually we'll hope to actually continue all this idea of utilizing bats as a way to improve our life. And his talk is going to be on bat-inspired targets to fight human disease. But before we go to the talks, I would like to ask you two questions. This is the tradition here. So, Professor Wang, what has been the most surprising scientific finding, and how did it impact your own work? Like, what is the direction that it actually affected you later on?
Yeah, thank you, Asia. So it's clearly, my career, I was trained as a biochemist, right? So has nothing to do with infectious disease or immunology, but, accidentally I got into Australia's institute to do animal viruses. So I think the most surprising discovery is that, we discovered the Hendra virus in Australia that really has a high case fatality for human and horses. And eventually we trace the origin of the virus and we discover that actually bats is the origin. So it's a very surprising discovery, but totally changed my career. But I think it beyond, changed the career of many other scientists and even the field, right? Immunology, for you guys, you would have never thought, you will have a bat immunology, as a model system. So that's, I think it's a single event, basically from virus that led us to bats. And then from bats, we try to understand more and more. And now the field is really open. And Matteo will give you very detailed examples of, how bats can be the new model for human medical studies. Yeah, thank you.
Serendipity, but not really, right?
Yeah, that's right. Yeah.
So, Matteo, can you describe the turning point in your career that defined your current research?
Yeah. Thanks. It's a great honor to share our bad lessons here. And for that question, I mean, there's a few turning point, but the most important one is actually the moment when I met the Batman here, Linfa. Yeah, so that was 12 years ago when I was still a MD medical student. So at that time, my dream was to be a neuroscientist and neurologist. Yeah, so but I met Linfa. Linfa gave a talk on bats. And I was so fascinated, and that I had so many questions. So I went to him and asked so many questions. We had a long chat on the same day. Yeah, so I think that really turned around, because that really convinced me that bats are very exciting. Well, those very high risks at that time to study bats as a medical student, but I was very convinced and I'm very excited, and I decided to study bats, and hoping that we can learn from bats to benefit human at one day. So I think I'm very happy that it's actually, I think it's happening, and I think we are just scratching the surface. So I'm very excited for the future discovery in the field of bats. Yeah, so that's very important turning point for me. Yeah.
People.
Thank you. Yeah.
Thank you very much. Yeah. And maybe let's go to the talk, and please, Linfa, if you can share your screen.
Yes. Okay. So you can see my screen now?
Yes.
All right. Okay, so thanks again for this opportunity. And as I said, it's a duet, so I will give a very brief introduction, then Matteo really get to the core of science that we have been doing, right? So I titled this as a bats and viruses, really to give you a journey that the bat immunology we're going to present today is really coming from the outbreak response that, I was personally involved. So we will touch, briefly on the bat facts, and the bat-borne viruses. Of course, SARS-CoV-2 right now is really hot, but I said, it's I use the terminology is a known unknown. And then there's a question on are bats special in terms of its ability to hosting virus. And lastly, to conclude is really, we start with bat-borne viruses, but now the lessons we learn from bats is way beyond viruses and infectious disease. And that will be the focus on Matteo's talk. So just start with the bat facts, the list can go on, but, the key things are bats are very successful mammal. They co-lived with a dinosaur and a dinosaur extinct, but they are now occupy 20% of all the living mammals on Earth. And then they have very unique biology features, but clearly the flying mammal is the only one, right? So they're the only mammal which can fly. In terms of the current research into medicine and health, these are the three really important features. They have a very long lifespan and a health span. A 7-gram bat can live up to 41 years. So imagine they have the similar body weight as a mouse, but mouse lives 2 to 3, right? And then they really anecdotally, we just could not find any evidence of they have a cancer, or at least very low frequency of tumorigenesis. Last but not least, I personally start this whole field is to identify bats as an important reservoir of a large number of viruses, and they carry this virus without symptom. Okay, so that's kind of the bat facts, when they're not fly, but it's even more impressive is during flight. Their metabolic rate can go all the way to 15 to 30-fold of the resting rate. And the heartbeats, can go all the way to 1,000 beats per minute, and body temperature is 38, 42 degrees. I always say, all these stress levels, if it happens to you as a human, we will not last, right? You will die, basically. Any of these will kill you. But for bats, these are very normal, and it happens every day because when they're flying out, nighttime, that's what they have to experience. So it's a very high stressful process of flying. And so, as I said, the whole journey of bat immunology really starts from studying the bat viruses. So I borrowed this terminology, really, the known unknowns and the unknown unknowns, really from, many 2 decades ago when people were just after 9/11, at that time, the defense secretary in USA, basically, he was the first one to use this terminology, right? They're the known knowns and they're the known unknowns and unknown unknowns. So basically to say that the unknown unknowns, just like terrorists, right? Bats are, the viruses are very similar like a terrorist. You don't know they exist until they really exert, their impact globally and on human health, just like COVID-19. So I have a very interesting career I don't have time to share with you because I never studied virology, never studied infectious disease, but accidentally got a job in Australia and I start to really get into animal viruses. And in 94, in Australia, we discussed the first virus that I eventually named as a hantavirus. And that was the really sort of, we call the first modern emergent zoonotic virus from bats. And then the last 25 years, basically my personal journey has been on one after another. Some are more major outbreaks like SARS, and the COVID-19. Then in between, we have a Nipah outbreak, the MERS outbreak, Ebola outbreak. We can argue about, how they originate and how they transmit to human. But one thing is clear, at least the ancestral virus of all these highly lethal viruses that cause human outbreaks are bat viruses, okay? So that's kind of being solved. And that they can happen anywhere, right? The hantavirus started from very clean island country like Australia. And then, we have the Nipah in Malaysia started, but now we have Nipah in India and other countries. And then, SARS-1 in Guangzhou in China, and then MERS in Middle East. And then, you have Ebola virus in Africa, and then COVID-19 back in Central China. So really no place is safe in the world, right? So the question, are bats special or different in the context of their ability to harbor virus? Anecdotally, we have quite a lot of evidence, to say, for example, you do challenges of any viruses in bats, they are able to produce clinical signs. And then we can isolate virus from totally healthy bat, cells, and the persistently infected in bat population, and a high level of viral prevalence if we compare with rodents, and also greater genetic diversity, especially for coronaviruses, and many of the ancestral lineages coming from the modern mammalian virus from bats. And the last really is a debate we had for a long time is that are bats a better reservoir? Do they carry more virus per species compared with other mammals? So this is a milestone paper, published in Nature and from the EcoHealth group led by Peter Daszak. Basically is that, they prove that bats actually carries more viruses, per species than any other mammalian orders, okay? And so that solved the problem of whether bats are special in their ability to carry viruses. But then, the next question is more difficult, right? What makes bats special? Is that genetic? Is that ecology? Are their immunity different? So that triggers really, our studies. And then, having studied this sort of field for more than 20 years. And then when COVID started, and then the Nature, journal really want us to really summarize the lessons we learned from this host. So if we put simply, we think the biggest lesson we learned is that bats among the different mammals, they are much better, equipped with control, what we call defense versus tolerance, right? So on one hand, in this review, you can go there, although it's 4 years old, but still, on one hand, they have the innate immune defense genes like the interferons, ISGs, H cell proteins, already at a high basal level. So we call them battle ready. So, and on the other hand, they have this mechanism to really make sure you don't overreact. So you can call this a tolerance, or overcome of a hyperimmune sort of response. And then, Matteo will go more detail in some of these pathways. So the virus-bat interaction is really now very, very interesting, a hot research area. Basically, I always say traditionally, right, as a virology and infectious disease, most of the research question you ask is why is a virus X pathogenic in host Z? Okay. But since we started, backbone virus, we started to ask the question the other way around. Why is virus X not pathogenic in host Z? Then, more excitingly, we can now go beyond to say, why is host Z able to prevent clinical disease upon infection by virus A, B, C, and you name it? Bats carry so many viruses in different families, and yet really difficult for the virus to induce any clinical disease. So as I said, we start with all the virus work, but since Mattei, as an MD student, work with me, and then we have many others, basically in the last, 12 to 15 years, we have gone beyond just study bats from an infectious disease angle. So we have gone beyond that. And then the question is like, it's a big question, right? Can bats teach us health lessons? Basically, translational from bat science to human. So I always joking about, it's very expensive. In 2013, I said it's a $10 million question. So I asked the Singapore government to give us $10 million, and we were successful. And the title of the grant is Learning from Bats for Better Control Viral Infections, Reducing Cancer, and then Living Longer. Clearly very ambitious, right? And then, 10 years later, we won another grant from the European Research Council, ERC Synergy Grant, highly prestigious, and carries quite a decent funding of around 12 million, euros. And so the title is Bat Protect. Again, learning from bats, a new strategy to extend the health span and improve disease resistance. Okay, so can we really translate, as Matteo was saying, really the bat immunology, 10 years later, within 1 decade, from very basic immunology studies, we identify the targets, in the inflammation pathway, and then there's a company now is formed. So that led to this very sort of lengthy news feature from Nature Biotech, and it has a very catching title, Bat Biotech Takes Flight. And basically says it's been a human dream for at least 100 years. It's a Nobel laureate, want to study, the gut microbes in bats and how to translate in human, but it would take another 100 years or so, until myself and Emma Tilling, we were the two really leading pioneer this field. And now there is a company, the Paratus Sciences, $100 million as a first stage investment, 100% dedicated bat research, and really try to identify new pathways, new targets to improve human health. And myself and Emma Tilling are very privileged to be on the funding sort of advisor for the company. So I will finish with, this perception in the media, right? When you're talking about bats, people thinking, oh, bats are bad, carries viruses. But I always say bats carries viruses. That's a fact, but it's not bats' fault, really. Have all these diseases, outbreaks. This is really human activities. Now, our study will go further to say not only that in the bat virus sphere, it's not bats' fault. Bat might be the savior that we can learn from bats to, resist disease and, improve our lifespan or health span. To prove that this is a hot area and it can translate into medical, benefits, I'm very proud. In Duke years, we only have like 60 MD PhD students graduate or be trained, and that's 7 from my lab. And consider myself is not a medical. I'm only a PhD, right? So I said that's an indirect evidence, but very strong to say this field has a bright future for human medical application. With that, I'd like to, really thank my team, and also many, many collaborators internationally. And thank you for your attention. And I'll pass this now to Mattei. Thank you.
Okay, thank you for such a great first half of the talk. Yeah, so I would like to continue about more details of what we have learned so far. And so the title will be the bat-inspired target to fight human disease, and that's exactly what I really hope to achieve about 11 years ago when I started the journey with Professor Linfa. Yeah, so I think I will keep the introduction very short, but I just want to highlight the key phenotypes that really attract us our attention and make us very excited to study them deeply. So I think I just want to highlight among all the bad facts. I think I'll highlight the three things here. One is they're the only mammal that can fly, and the other two are I would say more relevant to human health. So one is they get infected without getting sick. I call it antiviral disease phenotype. Then the second one is that they also live really long, and without all those age-related chronic disease that humans suffer. So they have very increased health span. So I call it anti-aging. So anti-disease and anti-aging. And so that really is very fascinating to me when I started the journey. But of course, initially we are more focused on the antiviral disease phenotype. So as Linfa already mentioned, bats really carry a lot of viruses, and many of them are very high impact viruses or highly pathogenic viruses to human and animals. But the most important thing to me as a medical student or clinician is that they can get infected, and in a few experimental challenge studies that the infection can be really bad, meaning the viral titer can be really high in the tissues or in the blood. But it's the lack of the symptoms or lack of signs of disease or lack of the immunopathology that is really striking to me. So I think there must be some certain follow mechanism that need to be uncovered. And the second thing I just want to show you one particularly very convincing graph on the right side is that the y-axis here is the longevity quotient, which is basically the ratio between observed lifespans and expected lifespan. So basically, it's normalizing the body size of all the mammals here. So this is 700 plus mammals. If you normalize the body size, they are mostly at the ratio of 1 here. So it's pretty proportional to their body size in terms of the lifespan. But I see all the blue dots here are bats who are clearly outliers. And another fun fact I always like to share is that there's 19 mammals that outlive human after normalizing body size. But none of the mammals, of course, have a clinician like myself or scientists like us or vaccine or hygiene, right? So, but these mammals outlive human regardless, and 18 of these 19 are bats. So I think statistically they are very convincing, but they are not just being lucky, right? Because bats actually have not really have very good lifestyle. Meaning, for example, in Singapore bats, they are eating 80% sugar every day without getting diabetes. And the flight actually, as the Linfa shares, is very metabolically demanding and stressful, and that actually can contribute to premature aging. But despite of all these risk factors or stress, bats are still living long. So this is really interesting mammal to study. And of course, initially, we due to lack of tools in this field, this very new field, about 10 plus years ago, we started to study bats from the basic things such as genomes and transcriptome. And we found some interesting findings such as positive selection in certain pathway, but no in-depth wet science at that time. And the initial wet science, we are focusing on interferon and antiviral activity. And we find some interesting findings such as high baseline, so-called battle-ready states, high interferon at the baseline. All these are very interesting findings. But again, this is not as universally conserved across bat species. And also, secondly, as I share, the very interesting part is not how they resist viral infection, but it's how they resist the disease upon infection. So that part is at that time is still not solved. And there's other interesting mechanism. They have elevated heat shock protein, which can help them deal with high temperature and stress, and also enhance genotoxin efflux pumps. And so all this I call it the upregulation of gain of function. So on the left side of this diagram, which we summarize in the Nature Perspective in 2021. And the part that I was really interested in for my PhD and the follow-up postdoc work is the down regulation part or dampen part, which is actually found to be very conserved and universally present in all the species. And then later on, we find that some of the lessons can be actually translated for human benefit. Yeah, so that will be my main focus for today's talk. So this inflammasome pathway is probably very familiar to most of the people in this audience, but I want to just quickly highlight the key things. This inflammasome is very broad. There's a broad spectrum of stimuli that can activate the inflammasome pathway in human or mice. That includes both microbial, such as RNA viruses, and also steroid signals, such as ATP and endogenous DNA, things like that. And I think this inflammasome, we think is very important at that time, despite the limited tools and understanding of their biology. It's because inflammasome has such a broad spectrum sensing, and bats, as we share, the most important unique phenotype, biologically, is that they are only mammal that can fly. So during that metabolic stress of flight, there will be a lot of injury, damage, and stress that could activate this pathway. And this pathway also increasingly recognized at that time that they play an important role in both infection and also aging and age-related diseases. Therefore, we think this is something pretty important to look at, and we believe that probably have an interesting adaptation. And quickly summarizing it, so we have this upstream sensor, M2 and NLP3 as example here. The middle part is adapter, and then they will aggregate into aspect upon activation, and it will recruit and activate caspase-1, the effector, leading to the pro-inflammatory lytic programmed cell descopraptosis. And also another thing that it does is the cleavage of the interleukin-1 beta or interleukin-18, leading to systemic inflammation. In contrast, bats have naturally evolved to dampen inflammasome, which is not surprising to me. But what is surprising to me was that they did it with multiple mechanisms at the different step of the signaling pathway. And that you can see here that I summarized in this diagram that we found 6 mechanisms just within this one pathway. They utilize to dampen this inflammasome pathway in bats, and you can see at all level, completely genomically lost. They at the genomic level, they completely lost this pi-hinged gene family, including M2. So this is at the genomic level. You can see on the right side, this NLP3, they have 2 different dampening mechanisms, and both at the transcriptional priming and also at the protein level, they have some mutation. And you can see downstream here, there's a specific mutation leading to disruption of the caspase-1 activity, and another set of mutation disrupt the interleukin-1 cleavage. Last but not least, we even find out there has a pretty gain of function negative. Yeah, so the first finding when I started as a PhD student is that with Linfa is that this genomic discovery. So this pi-hinged gene family are shown to be conserved in all the mammals, all the mammalian groups, right? But when we look into bats, all the bats we examined, at that time I examined 10 species. Nowadays we have examined much more than that. All of them have consistently lost this gene family. You can see here, I'm giving you just 6 examples of bats in boxes. Yin and yang they are 2 different suborders. And then you can see the more closely related species to bats, including this horse and dog. They all have this conserved red color pi-hinged gene family, right? The most famous one is M2, which is important inflammasome DNA sensor. And you can see clearly that all of them are lost in bats. So that was our first sign that give us confidence that the inflammasome probably is very different. But more exciting thing is when we did this experiment. But initially it was very frustrating because when we finally get some bat cells, primary bat blood cells from the Australian bats, we started to do some experiment by giving them all kind of inflammasome potent stimuli that are meant to kill the cells almost immediately within 1 to 2 hours. But whichever ligand we put the cells on, put on the cells, none of them work. So this I'm giving you 2 examples. ATP and adjacent, they are very potent NLP3 stimuli that can activate inflammasome and cell death within about 1 to 2 hours. And there's many other we try, of course, including DNA and other stimulation, including infection. But none of them can trigger cell death. First of all, they are not bothered by the stimuli, and also they don't have this inflammasome aggregation aspect. And you can see on the left, human cells. You can see nice aspect near the nucleus, which is basically aggregation of the S protein, which is really pretty hard to miss. But in a bat counterpart, it's really difficult to see. So initially we thought this is some technical error, or maybe the best cell quality is not good because we shipped from Australia to Singapore at that time. But after with consistent experiments, and this become the single most important negative result for me, because this is a foundation where we start to look into the mechanism why they don't have the inflammatory activation. Yeah. So in addition to the steroid stimuli or ligands, we also try viral infection. At that time, MERS is having epidemics. So we in the BSL-3, we did an infection to show that the human cells can have an inflammatory activation as expected at a dose-dependent manner. But bat cells, which is in red, there's no activation at all. But they have still pretty good infectivity, meaning that the virus can still replicate and infect the bat cells, but just they don't induce immune response. So this is again consistent with the special viral reservoir status. So to be a good reservoir, they has to be infected, but just they don't get the overimmune response that we get. Yeah, so this is a pretty consistent. Yeah, so that's lead to our my first wet sign paper in 2019. But it's very important for us for the first to discover the first mechanism, detailed mechanism for inflammation dampening in bats, with the implication for their special reservoir status. But I always said that this is a little bit late, but by then it's in US and Europe and all over the world, people already start to pay a lot of huge attention to NLP3, and billions of dollars already invested in this space to develop inhibitors against NLP3. Yeah, so that was a little bit late in terms of translation, but I think the later two examples give us opportunity to really do some translation and then to hopefully benefit humans. So the first one is this downstream mechanism. So when we put back M2, which is the DNA sensor that lost at the genomic level, but we still couldn't restore the inflammatory activation in bats. So we wonder, there must be some other mechanism at the downstream. So when we go down further with another MD, very talented MD PhD student, who is my junior, we figured that downstream is also dampened, but with very different mechanism. So as you can see here, it's just a simple diagram to summarize our finding. So at the time, we chose 3 different bat species. The reason is they behave pretty differently. So different from the previous two mechanisms, PyHin and NLP3, which are pretty universal across bats in terms of the dampening mechanism. But in this case, they have very species-specific mechanism. But the overall trend is the same, as summarized here. So Therapus electo is our Australian bat. For this Therapus electo, you can see that the caspase-1 activity is completely disrupted, but the ion beta cleavability is still preserved, right? So this is basically a caspase-1 dampening mechanism. And on the far right is maltodabidase, actually is from China. They chose very different mechanisms. So instead of dampening the caspase-1, they decided to mutate our interleukin-1 beta cleavage site, so they couldn't be cleaved. So they chose to dampen our interleukin-1 beta instead. And then the middle one is the Ionixurus Ionixurus is our Singapore local bats. They chose to do a little bit of both, a little bit of caspase-1 dampening, but not full disruption, and then a little bit of interleukin-1 cleavability disruption, but not fully. But overall, they achieve the same job, which is the dampen inflammatory at the downstream. And we always joke that the Singapore is always geopolitically neutral, as you can see in this diagram. Yeah. So among this discovery, we find out that there's a BET specific mutation in this Australian BET, Therapus lacto. And when we zoom in, the mutation is actually at the interface of the dimer of the caspase-1, the P10 dimer. And as some of you know, that caspase-1 inhibitor was pretty hot, and there are some inhibitors that go down all the way to clinical trial, but they failed the clinical trial due to the toxicity and due to potential off-target effect. And learning from this mechanism of dampening, which is not at the active site, but rather at the interface, we learned that whether we can learn from this mechanism to develop drug that can be targeting sites that are outside of active sites, such as allostatic site or interface. So we managed to design some BET-inspired drug screening assay with some exciting hits that we are doing some validation right now. And so this is the first lesson that's leading to potential translation. Now, the second one is the one that is a little bit more advanced in terms of the progress, is this S2 paper we published 2 years ago, where we showed that the BET S2, which is about 89 amino acid small protein, there is a pretty potent negative regulator of the inflammatory pathway. And the beauty of this is that they inhibit at the level of ASC, which is the first conversion of the signaling. And this diagram, I give you a few more examples of inflammatory cells. There are many more, but the point is that they all can all sense different stimuli. But eventually, most of them they will converge at converge at the level of ASC, and therefore by targeting ASC, you can block all the inflammatory activation regardless of sensor involved. And this is important because many human diseases, including the significant one such as atherosclerosis and neurodegeneration, initially people thought that NLP3 is the main culprit. But increasing evidence show that the multi-sensor involvement, that's the key word. So actually we need to target all the sensors, more than one sensor that are involved in the pathogenesis. Therefore, the best strategy here might be a wise one to target for those human diseases. So I think this is the second lesson we learned, and I filed IP for certain drug candidates, and then we licensed to the BET biotech company Linfa talk about. Yeah, and so I think the initial part that really struck me is that is there again in the unique features of bats in this context of BAS2. You can see that only primates and the bats consistently have the S2 gene on the genomic level. So this is another extreme example. But one thing I also want to highlight here is that a bat looks very different because of their wing morphology. But genomically or phylogenomically, bats are closer to human than mice. Therefore, we argue that bat could be a better model for translation, although it's more challenging due to lack of tool. But now we have much more tool to study, and we have multi-omics. So I think it's time for more bat research now. And so in addition to the genomic level uniqueness, we also find that at the mRNA level, endogenously, the S2 gene is highly expressed compared to the ASC counterpart, which is who they interact with. So that give me a confidence that this could be an interesting regulator that interact with ASC, and moreover, not only endogenous mRNA is high, when we overexpress it in the cells with the same mRNA expression using overexpression system, they still have more protein for BAS2 compared to human counterparts. So they have also post-transcriptional mechanism of enhanced protein level. So that's leading us to the most important functional study. So we started with this in vitro work. So we overexpress this BAS2 side by side with human S2 in different systems, including reconstituted human HEK293 system, and also more physiologically relevant THP1 macrophages system. And all of them show that the BAS2, in this case in red, show dampening inflammatory effect, but the human counterpart didn't show any inhibition effect. And if you look at the image here for on the left top corner, you can see that the S2, this is of course at a very high expression level. You can visibly see they form filaments instead of forming spike, and this filament of S2 will bind to the AEC and then prevent them from forming the spike that are crucial. They are crucial for downstream activation. Yeah, so that's in vitro evidence. Of course, give us more confidence, and then in order to validate this further, we made our first transgenic mice that contain a bad gene. We call it bad-type mice. Yeah, so for the first model we did in the context of infectious disease is that we give a lethal dose of influenza A virus in this baptized mice, and you can see the expression of BAS2 reduced the mortality by 50%, correlating with reduced lung inflammation, but without affecting the viral load. So this is another example consistent with their special viral reservoir status. So they can get infected, but they modulate the host response. In this case, dampen inflammation, leading to improved outcome. And to show that this is not specific to a virus, we also demonstrate with 3 different type RNA viruses, including the negative sense single-stranded RNA virus, which is influenza, which is shown in the previous slides. Zika at that time was pretty hot, and so the positive sense single-stranded RNA for this case. And the Malacca is a backbone virus causing some pneumonia in human patients. In this case, is a double-stranded RNA virus. So very different type of RNA viruses. So, but in all cases, they can activate inflammasome, and our S2 can dampen the inflammasome. So this is showing that this is pan-virus phenomenon and independent with the virus type. And of course, in the COVID pandemic, we also did experiment with SARS-CoV-2. But in this case, instead of activating inflammasome directly through the infection of cells, for this SARS-CoV-2, they tend to activate inflammasome through immune complex. So therefore, we designed this experiment to form an immune complex by coupling the RBD, which is part of the spike protein of the SARS-CoV-2 virus, and with the antibody against them, and then to form an immune complex. And then this immune complex can actually cause the clustering of the Fc gamma receptor 3A that lead to the inflammatory activation. Yeah, so basically you can see the immune complex can cause activation of inflammasome that can be inhibited by MCC950, which is one of the NLRP3 inhibitors. And also we can inhibit that with the S2 when we deliver them intracellularly, when we fuse them to the cell-penetrating peptide. In this case, example is TAT, cell-penetrating peptide, allow them to go into the cells to do the inhibition. Yeah, so in addition to the viruses, we also try steroid inflammation. So because inflammasome actually play even much bigger and broader role in the context of steroid inflammation and chronic diseases. So in this case, we deliver these monosodium urate causative agent for gout arthritis into the mice, and you can see our BAS2 lead to less immune cell infiltration and with less cytokine production. So they do also reduce steroid inflammation. Yeah, so these are some of the examples we did, and I think based on this discovery, we decided to target ASC with a different approach with a S2 as a prototype, and also we developed some drug screening assay, and we filed IP and license to this company, and we are very glad that they are willing to 100% focus on bad discovery and bad therapeutics, and we really align well in our mission, and we are working very closely on this project and also hopefully future projects. Yeah, so this is our first so-called big major lesson from bats, and basically anti-inflammation or regulation of inflammation that are leading to one of very important drug targets. And initially, of course, we are from the infectious disease lab, we will focus on viral disease, but we soon realized that the inflammation plays such an important role such a broad range of human diseases. Therefore, while I was practicing until last year, we had built a few local in Singapore, a few strong collaboration with clinicians, and we are evaluating inflammasome and the markers, and also preclinical testing in different diseases, including neurodegeneration, atherosclerosis, and then other example as shown here. And also I have ongoing development. I mentioned briefly about Cas1 and other inflammasome or inflammation-related drug targets, and ongoing translation and commercialization. Yeah. So I think that's the main message I hope I want to share. I think this is just one example. I think we have learned from that so far, but I think we believe this is just scratching the surface. It's because the reason I say that is when we knock out inflammasome, inhibit inflammasome in mice, which we call it bat-type mice, they are no way close to the bats, right? Bats on average living 3 to 4 times longer, and some of them living up to 10 times longer with very good health. And those mice that we baptize actually only maybe about 5 to 10% longer, and a bit of better health due to dampen inflammation. So there's still a huge gap of understanding why they are special, especially in the context of aging and anti-disease. So that's our lab mission. We really want to make use of the resources in Singapore, which we built by Linfa. Since 10 years ago, we have a bat colony. We can do a lot of in vitro and even in vivo studies. Now we are also tapping into the omics platforms, and we can do a lot of studies and comparative studies, immunology, and also beyond immunology, such as metabolics, and even neurodegenerative studies. And we can translate those findings to specific mechanistic studies and projects, and eventually the goal is to find out the secret behind their healthy longevity. And we believe that while we are trying to, in the current globally aging society and the global aging kind of phenomenon, while we try to figure out a better way for healthy longevity, we believe that nature, the bats already have solutions, and we are very excited to discover more from bats and more inspired by bats, so that we can benefit humans. And yeah, so with that, of course, my special thanks to Professor Linfa, who really pioneered this and convinced me to do this with him. And we are with a very good backpack who have done all the work, and all the collaborators and the funding body as well. And with that, special thanks to the bats as well. And yeah, and it's a really great honor to share our bat work and bat lesson with all the global IMMUNOTALK community. Yeah, thank you so much.
Thank you very much. And it was, I'm sure there are going to be so many questions, because I know that I already have several ones. So you can see here that you have several ways of asking questions, all of them online. The Q&A session is not live, so you can post your questions, and you will get hopefully a very lively discussion online. I also want to give a shout out to our next week seminar, Global IMMUNOTALK by Ziv Schulman, and which I'm sure is going to be a very interesting talk as well. So with that, I would like to thank you again for joining us at this late time of the day and sharing with this really amazing insights and new way of looking at immunology. So thank you very much. Thank you all for listening, and good night.
Thank you.
Thank
Bye-bye.
Thank you for inviting. Thank you.